This course is approved by the AAFFP for 2 credit hours. And it's Module 6, Validation and Verification of Laboratory Test. So what is test validation and verification? Process that establishes that a new test method or system has performed as intended. It focuses on accuracy, precision, sensitivity, specificity, and other performance metrics. And verification. This process confirms that an existing test method works as expected in a specific lab setting. It ensures consistency with the manufacturer's claims and established performance standards. So validation is going to be typically when there's a new system, or laboratory developed testing, and then verification is when there is a test that has been gone through extensive validation in another lab setting, but you're testing out how it works in your laboratory setting. So an FDA approved test that has been implemented into your laboratory, you might be performing you would only be performing a verification instead of a full validation. Whereas an LDT, or laboratory developed test, is a full validation. The purpose is to ensure that tests provide reliable, accurate, and reproducible results, both validation and verification. That's the purpose of them, and to meet regulatory requirements for quality assurance under CLIA. They both have the same purpose. There's slightly different applications, however. For validation, it's required for new test systems or modifications to existing tests. So if you're implementing a new test system, a new laboratory developed test, a new LDT, or if you're making modifications to an existing LDT or an existing FDA test, modifications require validations. You need to show that whatever changes or if you're having a new test that's laboratory developed, you need to ensure that it meets all the requirements. However, verification is needed when adopting a test system that's already been validated by another organization, such as FDA approved test. So FDA, one of the regulatory bodies, the Food and Drug Administration, if you remember from lecture from module 1, they develop test systems and complexity, or they delineate complexity. So they approve the developed tests. So there's a very extensive procedure to get a test system approved by the FDA. That's why if you're using an FDA approved test, then you only need verification because it's already gone through extensive validation under a certain set of parameters. So oftentimes, the reagents, whatever it is, um, you can't really change the reagents. You have to specify what the manufacturer's guidelines are, follow the recommendations, follow everything. And if you follow everything properly, then you only have to do a valid verification instead of a full validation. So the principles of validation. What are the core validation metrics? Accuracy. This shows that the result aligns with a true value. Precision. Consistency under identical conditions. Okay, so can you repeat and get the same result? Sensitivity. Correctly identifying true positives. Specificity. Correctly identifying true negatives. And reproducibility. Consistently across operators, equipment, and locations. This is also called ruggedness, meaning reproducibility or ruggedness. Precision is often called repeatability. Just nomenclature, good stuff. So the validation phases. You have a planning phase where you define objectives and performance criteria. You identify necessary materials or controls that you need for your validation. Then you execute that plan. You conduct experiments to measure performance metrics, collect data systematically, and then move on to the analysis phase. So once you collect all your data, once you perform all your experiments, then you analyze your data and you evaluate your data against your predefined criteria. So it's important to define your criteria before with your validation plan. So you should have your target. You should know what you're going to try to achieve before you start your validation. It's an important metric. and then identify any potential limitations or anomalies. So the whole purpose of validation is to show that your system is working properly, but you can also identify any limitations. If there are limitations, that's not a problem. As long as you have it documented and you know what your limitations are, you're not going, when you're testing patient samples, you're not going beyond the limitations of your assay. Every assay has limitations. So some key considerations. Establish performance thresholds. Is often based on regulatory guidelines, but you should consider clinical relevance. So not everything has guidelines, not everything has a regulatory guideline. So you should be basing your thresholds on clinical relevance. So say in toxicology, you should not be trying to have a limit of detection, well below and then have an analytical range that doesn't make sense for your analytes. If it's in an amphetamine that has an average concentration of 1,000 nanograms per ml in urine, and when it's being actively used, then it doesn't make sense to evaluate from 1 nanogram per ml to 10 nanograms per ml, because then you're just gonna have numbers well outside your range. And then it also doesn't make sense if fentanyl is very low concentration in the body. It's 0.1 nanograms per ml in blood to 10 nanograms per ml, then it doesn't make sense if your LOD is only 100 nanograms per ml, and then your range is 100 nanograms to 1,000 nanograms per ml. That doesn't make sense. So it's important to have clinical relevance. Use controls and standards. This helps minimize variability. It's always important if you use controls and certified standards, then it's going to reduce your variability. If you're using in-house controls or patient samples to try to do your validation on, then you're going to have a lot more variability than is if you use controls or standards. There's nothing wrong with it. You can technically, for some parts of the analysis, you can use patient samples, but you're going to increase your variability and you're going to increase um, your difficulties if you do that. And document your processes and outcome. Always be transparent, and this helps enhance reproducibility. Why this helps reproducibility. So if you're transparent, if you're documenting everything and doing what you're supposed to be doing and showing what you're supposed to be doing, then it's going to enhance your reproducibility because it can be reproducibility, remember, it's through different test systems or different operators. So if you have a thorough documentation of the process, then your reproducibility is going to be better because then more operators can do the same thing. If one operator knows what they're doing because they know what they're doing, not necessarily what the because what the SOP says, or your documentation says, then you're going to have a worse reproducibility, especially with newer techs who aren't as exposed to um, things like that. So it's important to establish your performance thresholds, minimize your variability where possible, and document. What are some key differences? For validation, this confirms that a test method performs as intended for its specific use. This typically applies to newly developed tests or those adapted for novel purposes. On the flip side, verification confirms that an established, validated test performs as expected in the specific lab environment. So basically, an FDA approved test here in the US, it's FDA approved. There's not a ton other organizations that are approving. There is a kind of um what's the call? There's an approval body for New York. There's a criteria and regulatory body that you can do your validation, you can send them your data and things like that, and they'll kind of, I guess, what's the word I'm looking for? They'll give a stamp of approval that your study is rigorous enough and it meets the regulations to be deemed a verified test. But for the most part, typically in the market, it's an FDA approved test. That's what they're going to tell you. FDA approved means that it might be moderate complexity. It could be FDA approved and high complexity. But when they're FDA approved, then you do not have to have the same strict validation compared to verification. So what is the scope? Validation is a comprehensive assessment of all performance characteristics, including accuracy, precision, sensitivity, and specificity. Also, stability is something that you can keep in evaluated by validation. Whereas verification focuses on reproducibility and alignment with manufacturer's claims or external validation studies. A verification oftentimes is not they do not require the same ruggedness, the same thorough investigation from the lab because the manufacturer did a thorough evaluation and they sent that they sent their data, they sent their claims to the FDA. The FDA goes through a long process and says, okay, you just say everything you say is true and everything that you claim is approved. On inside those manufacturers, so they have a packing slip is what, or a package insert that we often call it. It's called intended use. And then that has a sheet. That packet has information on it. It has sample stability, so it has how long the samples are good for under room temperature, how good they're under refrigerated, how good they are frozen. So the manufacturer for this assays evaluated the stability thoroughly for those analytes. Also, interferences on the package inserts to list any interferences that may have been there. That impacts the specificity and sensitivity of the assay. Um, they do that thoroughly. They dig through all that stuff. They identify any interferences, any issues with matrix effects, things like that. And they thoroughly go through that so that way when you do your verification, you're mostly looking at reproducibility, which is mostly going to be your accuracy and precision. Whereas a validation, full validation, you have to do that stuff. You have to do the accuracy, you have to do the precision, you have to do the sensitivity, specificity, you got to do your LOD sensitivity, you have to do your specificity. Any interferences, you have to do your linear range if you're doing a quantitative assay. You have to do um any kind of interferences, cross reactivity, carryover, things like that. You have to do all that stuff. So the validation is way more intense because for the verification, someone else already did the heavy lifting. These FDA approved kits are more expensive because they did the heavy lifting, so they pass on that expense to the user, as always. So some examples. I think I just gave away all the examples. When relevant, a new assay for a non-standard biomarker. Or if you're modifying an FDA approved test for one used on a different platform. So some examples, developing a new assay for a non-standard biomarker. Maybe you're developing an assay for a blood biomarker for some cancer. Maybe that's not been evaluated by the FDA. They don't have that on the market, but you evaluated it and you found some good markers, or you found a paper that's published on a marker, but no one's evaluated it and studied it on an instrument or your instrument or your immunoassay or whatever you're doing. So then you have to do the full validation on it. Or if you modified an FDA approved test for use on a different platform. So there's a bunch of different platforms out there, even for just toxicology screening instruments. You have your Siemens Vivo Pro, you have your AU systems, you have Abbott systems, you have Beckman, um, all these things. If you want to use a reagent, say you have a Vivo and you want to use Abbott reagents, so you have a Siemens instrument, but you want to use Abbott reagents, oftentimes that's going to upgrade you from, even though the reagents themselves may be FDA approved, the Abbott is FDA approved on the immunotox, but it's not FDA approved on the Vivo Pro. So then you have to, if you decide you want to use your immunotox reagents on a Vivo Pro, it becomes a modified FDA test, and then you have to do a full validation. Yeah, fun stuff. As whereas verification is when you choose an FDA approved test in your laboratory, and then you're just verifying the performance of the new batch of or verifying new batch of reagents or equipment. So if it's the same we do lot to lot verification. So if you get a new lot of reagent, oftentimes you'll do a quick verification where you do like 10 injections to evaluate the new lot. Compare that to the old lot, and you're just comparing accuracy and precision. That's what oftentimes verification is just accuracy and precision. Typically, that's what you're evaluating. Um, so in this instance, if you want to use, you want to use amino analysis reagents, you want to use Abbott reagents, then you need to get an imitox. You get an imitox and their instrument, and that's on approved. But if you want to use the Abbott reagents on a Vivo Pro, then that becomes a validation. So the same kit, the same reagents, depending on the platform, the instrumentation that you're using, it could be the difference between having to perform a verification or performing a validation. Also, verification, moderate complexity is oftentimes a verification. Moderate complexity is going to be an FDA approved test. It's not going to be a laboratory developed test. So moderate complexity verifications, high complexity high complexity LDT, anything that's an LDT, even like if they say that you hear LDT, it's going to upgrade it to high complexity. High complexity means you're going to do validation, full validation. So that's the difference. That's also a difference there is that I just think that's what the slide was going to be. CLIA requirements. Both require are both required under CLIA. Um, applicable for high compliance testing. The level of vigor varies between the two tests. And there are different documentation standards. Typically, by different documentation standards, what that means is there are different aspects that need documentation. There's different experiments that you need to document. So like LOD, LOQ, sensitivity, specificity, those would be required for validation, whereas accuracy, precision is required for both. But that's where you stop for verification. So validation is an exhaustive process required for new methods, focused on ensuring test methods meet intended user needs, while verification adapts existing methods to specific laboratory conditions and ensures accuracy within the lab's unique environment. Both processes are governed by regulatory bodies like FDA and CLIA and involve documentation, compliance, and continuous monitoring.